Today I will discuss another gram-positive rod, and that is Corynebacterium diphtheriae. These are your learning objectives. However, you need to focus more on pathogenesis and laboratory diagnosis of Corynebacterium diphtheriae. Let's have a quick recap of gram-positive rods classification. So Corynebacterium is non-spore forming and is aerobic. This is the brief introduction of Corynebacterium diphtheriae. So it's a gram-positive rod. It is non-motile, it is non-spore forming and non-capsulated. So try to remember this with MSc and just put 3 N's. It has 3 biotypes: gravis, intermedius and mitis. Corynebacterium diphtheriae produces pharyngeal diphtheria, which is common in children, and it can also produce cutaneous diphtheria. It is mostly common in unvaccinated or immunocompromised individuals. Humans are the only natural host. This organism resides in the upper respiratory tract. The mode of transmission is mainly through respiratory droplets, either by coughing and sneezing. In case of cutaneous diphtheria, it can also infect skin at the site of pre-existing skin lesions. And this is primarily common in tropics, those who have poor skin hygiene. Pathogenesis of Corynebacterium diphtheriae. So there are 2 types of strains, toxigenic and non-toxigenic strain. Please remember, this non-toxigenic strain is not lysogenized. It will not produce diphtheria toxin and hence it is not pathogenic. Whereas this toxigenic strain, it is lysogenized, it will produce diphtheria toxin and hence it is pathogenic. So we'll talk about this toxigenic strain. Temperate bacteriophage attaches to bacterial cell and it merges its genome with that of bacterial genome. This bacteriophage has tox gene and this actually codes for diphtheria toxin production. So following this, Corynebacterium diphtheriae can make diphtheria toxin. Now let's have a look at this diphtheria toxin. It has 2 subunits, A and B. B has attached to human cell with the help of receptor, and both of these have endocytosed and they are engulfed by cell membrane and an endosome is formed. Medium inside this endosome becomes acidic and both of these are actually joined together by disulfide bonds. They break and both of these subunits, they separate. This A subunit is actually enzymatic and it has ADP ribose group which attaches to elongation factor 2. Please remember, this elongation factor 2 is an important ribosomal protein that joins amino acids together during protein synthesis. So once there is ADP ribosylation of elongation factor 2, there is inhibition of protein synthesis and this leads to cell death. So once this Corynebacterium diphtheriae enters into pharyngeal epithelial cells, as I mentioned before, after multiplication there would be exotoxin production. So this can end up into local inflammation and there is tissue necrosis, and this results in the formation of a pseudomembrane. So pseudomembrane is actually, it's a combination of fibrin, bacteria and inflammatory cells, and it becomes a leathery adherent membrane. If this can extend into larynx or trachea, so it can produce mechanical obstruction, respiratory tract obstruction, And if it penetrates deeper into pharyngeal wall, this toxin can be absorbed into bloodstream and there is toxemia. Now this toxemia has affinity to myocardium, to nerves, and we'll see what are the complications that can arise from this toxemia. Clinical features of pharyngeal diphtheria. So there is formation of a pseudomembrane. It's a tough gray adherent membrane. There is fever, sore throat, and cervical lymph nodes. They can enlarge, and this bull neck appearance is a characteristic finding in case of pharyngeal diphtheria. If it is cutaneous diphtheria, mostly the ulcers, they are shallow and they can be covered by grayish membrane. Complications of pharyngeal diphtheria. As I mentioned before, if this membrane extends into larynx and trachea, this can cause airway obstruction. If it reaches heart, so it can result in myocarditis, and this can produce arrhythmias and even heart failure. It can also cause nerve weakness or paralysis, especially of the cranial nerves. Paralysis of muscles of soft palate and pharynx can lead to regurgitation of fluids through nose. There can be peripheral neuritis, which actually affects the muscles of extremities. So there can be nerve weakness, or you can say paralysis and peripheral neuritis. So what are the complications? Number 1 is airway obstruction. Number 2 is myocarditis. Number 3, nerve paralysis and peripheral neuritis. Lab diagnosis of Coryenbacterium diphtheriae, day 1. Just think of clinical features and you can write down different specimens. Throat swab. Okay, there can be nasopharyngeal swab, there can be skin swab, and for all of these swabs, you can transport this into sterile serum. And on performing gram staining, you will find gram-positive rods. Now, the gram-positive rods, they have a particular arrangement in this case, and this is known as palisade arrangement. If you can see this palisade arrangement, they are in the form of V-shaped, they are in the form of L-shaped. So you can see this L-shaped and V-shaped arrangement of these gram-positive rods. So they are also club-shaped, wider at one end. Albert stain. It's a special type of stain which is used for staining the volutin granules, or also known as metachromatic granules. They are found in Coryenbacterium diphtheriae when they are grown in a loveless serum. So large number of granules can be observed by this Albert stain. So they are green colored bacilli with bluish blackish metachromatic granules. They are at polar ends, and once they are in the form of clusters, they give Chinese letter pattern arrangement, or you can say they give beaded appearance. And these beads, they actually consist of these metachromatic granules. They are storage mechanisms for high energy phosphate bonds. Methylene blue is also a good stain. The metachromatic granules, they take bluish purplish color with this methylene blue stain. Okay, so we are continuing with day 1. So apply this on blood agar, and then we have few selective media: Telluride blood agar, Loeffler serum medium, Tinsdale medium. On day 2, observe colonies on blood agar, they would be just gray colored colonies. And if they are grown on this telluride blood agar, so they would be blackish colored colonies, as you can see over here. This is because potassium telluride, it reduces to potassium tellurium salt, and colonies, they have blackish color. Three biotypes on this blood potassium telluride agar. In case of gravis, these colonies are gray, raised from center, and they have crenated edges, and they look like daisy head. If we talk about mitis, mostly it has shiny black convex and smooth colonies, and they just look like poached egg. If you talk about intermedius, again it has gray, black, dull granular center, and it has lighter center near edges, and it looks like frog's egg. Loughless serum. It is composed of nutrient broth, glucose, and serum, and Coryenbacterium diphtheriae would produce small, circular, cream-colored, glistening colonies with irregular edges. In case of tin stale medium, grayish-blackish colored colonies which are raised, and they are surrounded by, as you can see over here, they are surrounded by dark brown area, and this is because of H2S production from cysteine, which interacts with telluride in this medium. On performing gram staining, again you will find gram-positive rods, and you can see the V or L-shaped arrangement. They are catalyst positive, non-motile, go for biochemical tests and sensitivity. Now, in case of biochemical tests, there is a test which is known as Hiss's serum sugar test, and it consists of glucose, maltose, sucrose, and starch. So once there is acid production following sugar fermentation, a distinct yellow color is produced, and this is because of phenol red indicator is present inside these tests. So day 3 will result interpretation day, and you will interpret the Hiss's serum sugar tests. You can find glucose, maltose, there is acid fermentation. So for all of these three biotypes, starch in case of gravis, and it is negative for mitis, for intermedius. The acid fermentation is also negative in case of sucrose for intermedius, and this is variable in case of sucrose for gravis and mitis. Read the sensitivity, and alternatively you can also apply API Coryenbacterium. Now we will talk about virulence tests. So the first one is Schick test, and it has epidemiological importance, and it checks the immune status of person. So intradermal injection of 0.1 ml of toxin, and after that, if no antitoxin is present in this person, there will be inflammation at the site in about 4 to 7 days later. If no inflammation is there, It means antitoxin is present and the patient is immune. Another virulence test, and it is known as Ilex toxigenicity test, and the aim is to find out toxin of Corynebacterium diphtheriae. Soak a strip of sterile filter paper in diphtheria antitoxin and lay it in a petri dish. Pour 20 ml of sterile rabbit serum agar onto this strip. Let it cool and then inoculate test positive and negative control organisms. So this is positive control stain which produces toxin. This is negative non-toxigenic control strain, and this is test strain. Incubate this overnight at 37 degrees centigrade and look for lines of precipitation. So these are lines of precipitation. The toxin produced by bacterial growth diffuses into agar, where it meets the antitoxin and then it produces lines of precipitation. So it means that this test strain is toxigenic. Here there are no lines of precipitation, so this is non-toxigenic. Here there are lines of precipitation. As you can see, these are also lines of precipitation. If you can see over here, lines of precipitation. So all of these are positive for diphtheria toxin. Alternatively, there is also PCR for toxin gene in Corynebacterium diphtheriae, which can be done. So summary is Corynebacterium diphtheriae produces pharyngeal diphtheria and cutaneous diphtheria. There are club-shaped gram-positive rods with palisade arrangement. There is a special stain, which is Albert stain, in order to see metachromatic granules, which are actually not spores, they are just granules. And there is a lysogenized diphtheria toxin, which is pathogenic. This is your assignment. Thank you.